Hello students, a very warm welcome to the official page of Read My Course. I am your biology teacher Dr. Shatabdi, and today we will be continuing with cell, a unit of life. Today we will be studying about two important structures in an eukaryotic animal cell, that is centrosome and its constituents, that is centrioles. So let's start. First of all, we will have a look at what are the topics we are going to cover today. We will look a little bit into the discovery of centrosomes. Then we are going to go into what are they, what are centrosomes, what are centrioles, what are the different parts of a centriole, and then finally we will end our session with a detailed discussion about the various functions which are performed by a centrosome and what is its important role in cell division, right? So let's start. In the first slide, I want you to have a very good look at the cell structure. See, in the cell structure, what we are noticing is that we have just around the nucleus, have a good look, just around the nucleus, we are seeing something which is known as centrosome, right? This round circular structure. When we are doing a magnification of the cell, then what we are finding is that a centrosome looks somewhat like this. That is, what does it have? It has two perpendicularly shaped, one like this, one like this, two perpendicularly placed structures, cylindrical structures, this one and this one. These two are the centrioles, right? They are the main constituents of a centrosome. And then we find that it is surrounded by a proteinaceous matrix which is known as the pericentriolar matrix, PCM, pericentriolar matrix. So these centrioles and the pericentriolar matrix together, they form what? They form the centrosome. So the centrosome is the cell organelle which is found in a eukaryotic animal cell. Remember guys, it is very much exclusively found in animal cells. It is not found in plant cells. This is the first important thing which you need to remember about centrosomes, right? So it is found in animal cells. It consists of two centrioles. These centrioles are nothing but cylindrical shaped structures which are arranged at right angles to one another, and surrounding that we have a proteinaceous matrix structure which is known as the pericentriolar matrix. Together this matrix and the centrioles form the centrosome. Okay, when we give a more detailed view to the centrioles, what we find is a structure like this. These are cartwheel-like arrangements, as we can see over here. These are cartwheel-like arrangements and they are arranged perpendicular to one another. These cylindrical structures, these individual cylindrical structures that you are noticing, tube-like structures which you are noticing, they are the microtubules. They are the microtubules. So microtubules are the structural units of a centriole. Okay, microtubules are the structural units of a centriole. And basically these microtubules are made up of a protein which is known as tubulin. This is the main composition. Other than that, apart from that, we have other proteins also, but remember that the main protein is tubulin protein. Okay, so with that we move to the next slide. This is the structure of a eukaryotic animal cell, as you can notice. This is the this is the nucleus. Just around the nucleus, just beside the nucleus, we find this red structure. This is the centrosome, and these T-like structures, we find two T-like structures, two structures forming a T, those are the centrioles inside the centrosome, right? And another important thing that we see over here are these filaments, fibers which are extending, right, throughout the cells. What are these? These are the microtubules. These are the microtubules, right, which are spreading throughout the skeleton of the eukaryotic cell. Another important thing that we need to remember about this structure is that it is not a membrane bound structure. Unlike other cell organelles which are normally membrane bound, centrosomes are not membrane bound organelles. Okay, with that we move to the next slide. A little bit about the discovery. It was discovered by a Belgian scientist in the year of 1883, and his name was Edward van Beneden, right? This is the picture. See, this is the picture of the scientist who discovered centrosome, that is Edward van Beneden. Then the name centrosome was not given by that particular scientist who discovered it. The name was given later by a person who was known as Theodore Boveri. The name centrosome was coined by Theodore Boveri. This is the picture of Theodore Boveri. With that, we move on to the next slide. So by now we have understood what are centrosomes. It is an organelle which serves as the organizing center of all the microtubules in an animal cell, right? We have understood this much. Now it assembles all the microtubules into a spindle during cell division. We will go into this in the later parts of the session. So remember, for the time being, just remember this much. It is exclusively found in eukaryotic animal cells, we have seen that, and it acts as a center for the organization of the microtubules, right? So what is the location of a centrosome? It is located just next to the nucleus and it is attached to the nucleus. So when we are having a electron microscopic view, when we are having an electronic microscopic view, we are finding a picture somewhat like this. See this cartwheel like structure, this is the centrosome. It is a very prominent structure which can be seen very easily under a microscope. Okay, and the basic features which we find in a centrosome are, see, we have a mother centriole, we have a daughter centriole. Now you can ask me, what is a mother centriole, what is a daughter centriole? We know that there are two centrioles, right? The mother centriole is the original centriole which is present, and the mother centriole induces or leads to the formation of a new centriole. This new centriole is the daughter centriole. So when does this happen? When does the mother centriole give rise to a daughter centriole? It is basically during the time of cell division. It is during the time of cell division that the mother centriole gives rise to the daughter centriole, right? Then we have the pericentriolar matrix, which is a proteinaceous base. We already know that, and we have the microtubular structures extending from the center towards the periphery of the cell, right? We understand that this whole structure together is the centrosome. Okay, with that we move to the next slide. Yes, centrioles. So centrosome is an organelle consisting of two cylindrical structures which are the centrioles, right? Now we know that centrioles lie perpendicular to one another, tubular structures. Now when we see the cross section, when we cut a cross section across a centriole, what we find is a cartwheel like structure like this, a cartwheel like structure. What are these three units consisting of? See, three units, right? There are 9 such units. There are total of 9 such units, each consisting of triplets. Okay, so that is the 9/3 arrangement. 9 such tubules consisting of triplets. The triplets are composed of what? Those triplets are composed of these tubulin proteins, alpha and beta tubulin proteins. We have known that, right? Now these adjacent triplets are again linked to one another. See these black bonds existing between the triplets? This is the link between the triplets. So in one word we can say that they are a 9 cluster of 3 fused microtubules. 9 clusters of 3 fused microtubules in each. This is the basic structure of the cross section of a centriole, of what a centriole looks like when we cut it into a cross section, right? Next we move on to the next slide. See what are the different parts of a centriole. Now when we cut the cross section, we find a central portion which is the hub. This is the central hub of a centriole. From that we see these spoke like structures radiating out. These are the radial spokes. Okay, then we have A, B, C, these 3 triplets, right? These are the arrangement of the microtubules. Okay, we have already studied, we find the link between those triplets. This link is what? Nothing but the AC linker. AC linker. So that is it. We have a central hub, we have radial spokes, emerging from it. Then we have those triplets A, B, C. Those are the microtubular triplet structures, basic structures, and those triplets are joined together by links, AC links. Okay. So centrioles are found only in animal cells. We already know that. They are made up of protein strands which are known as microtubules. We have studied that. Centrioles are made up of 9 triplets of microtubules arranged in the form of a cylinder. We have studied that. Now around these centrioles, what we have? We have studied, we have PCM, pericentriolar matrix. Now what is the constituent of this pericentriolar matrix? It is made up of proteins like tubulin, we have known. Two other, remember, one is pericentrin and ninine. Okay, one we know, tubulin. Two others are pericentrin and ninine. These are the proteins which are present in the PCM. With that, we move to the next slide. Yes, the functions. The most important function which is done by a centrosome comes during cell division. Okay. For that, we have to know a little bit about the various stages of cell division. We have not gone into the details of cell division, mitosis, meiosis. We don't know about them right now. So I will try to explain to you only the basics right now. When we will study the cell division in details, then we will go into a little bit again about this function of the centrosome, right? So what we know till now is that the humans have 23 pairs of chromosomes, that is 46 chromosomes that we know. So basically, in order to maintain all the genetic information intact in a particular cell, when a cell is dividing, then that daughter cell also has to indulge in keeping the 46 chromosomes intact, right? So that is where the role of this centrosome comes in. The centrosome is, the most important function of a centrosome is to maintain a equal, just have a look over here. Its function is to maintain a equal distribution of chromosomes in the daughter cells. Okay, so the function of centrosome during cell division is to maintain the chromosome number, that is 46 will remain 46 when it is multiplying from the mother cell to the daughter cell. Okay, how it does that we will see. Another important thing, remember that the centrosomes in the mother cell transforms into the nucleus of the daughter cell. How this happens, for that we need to move on to our next slide. See, in a human body, cells divide by two processes. One is mitosis and the other is meiosis. Most of the cells divide by mitosis except the reproductive cells which undergo a process which is known as meiosis. So for the time being, we don't have to read about meiosis, just leave this portion out. We have to read about mitosis. Okay. So what is the main difference between mitosis and meiosis? See, in mitosis, a cell gives rise to diploid cells, right? That is, they maintain the chromosome number. But in case of meiosis, what happens is they give rise to haploid cells. That is, the number of chromosomes become half. That is the basic difference, okay? We have cell cycles for both these processes. We don't need to go into details about them right now. Remember that in mitosis, the chromosomal number remains intact. In meiosis, the chromosomal number gets halved. So you will ask me a question that how is it that in mitosis, in spite of the cell dividing, in spite of one cell dividing into two daughter cells, how is it that the genetic material is not dividing? How is it that the genetic material is intact? Number of chromosomes is intact. That is because of the presence of these centrosomes. Okay. So let's go to the next slide to understand it a little better. See, in case of mitosis, we have some phases, we have some cycles. Okay. The first phase is the interphase. Then we have the prophase. Then we have the metaphase. We have the anaphase, and then we have the telophase. And that is the whole cycle. Okay. We start from the interphase. Then we have the prophase. Then we have the metaphase, anaphase, telophase. Right. And then a single cell divides into two cells. Now, what is the role of the centrosome over here? See, you will appreciate the fact that in interphase, what is happening? See those two red shaped structures at the top, that is the centrosomes. What happens is the centrosomes duplicate, that is the centrosomes divide. Okay. So, one cell has 2 centrosomes right now, 2 centrosomes, and each centrosome has 2 centrioles. That means a total of 2 centrosomes with 4 centrioles in one cell. During the interphase, this happens. Then as the cell enters the prophase, which is the second phase of the cell cycle, what happens is those 2 centrosomes, they start moving apart, they start drifting apart, where one towards the north pole of the cell, another towards the south pole of the cell. Okay, see, they are drifting apart. And another thing, you will be able to see these green structures in between them. You know what are they? They are the spindle fibers. They are the spindle fibers, right? Next, what is happening, see, in the metaphase, these 2 centrosomes, they have reached the opposite poles, one at the south pole, another at the north pole. Okay. Another important thing that we are noticing right now is that from the interphase where we find these thread-like structures, which are the chromatin, right? They are the chromatin fibers. Okay, we find they have become well organized into these X-shaped structures we had studied in our earlier class. They are the chromosomes. So the chromatins, they arrange themselves, organize themselves into chromosomes. Another important thing that we are noticing is that this nuclear envelope has gradually vanished over here. From the interphase, as the cell is gradually entering the prophase and then into the metaphase, the nuclear envelope is disintegrating completely. There is no nuclear envelope. Okay, the chromosomes become free, right? Then what happens is the chromosomes arrange themselves around a midline, that is the equatorial plane, that is known as the equatorial plane. And what we are noticing is that from these centrosomes, these spindle structures are getting attached. They are getting attached to the chromosomes. They are getting attached to the center of the chromosomes. We have already studied that the center of the chromosome is what? The centromere, right? The center of the chromosome is the centromere. So they are getting attached to the centromere of the chromosomes. Now comes the anaphase. During the anaphase, what we are seeing? We are seeing each chromosome has divided itself into 2 sister chromatids. Chromatids. What are the chromatids? The chromatids are the 2 identical arms of a chromosome. Okay. So the one chromosome has divided itself. See, the chromosomes have started dividing. The chromosomes have divided themselves into equal parts, that is the chromatids. The sister chromatids are gradually moving apart from one another. They are getting split from the midline and they are getting apart from one another. Why is this happening? This is happening because of this pull from the centrosomes. The spindle apparatus which is originating from these centrosomes. See, the spindle apparatus, they are pulling apart the sister chromatids, one towards the south pole, one towards the north pole. Okay. Finally, in the telophase, what we are noticing is that there is a complete separation of the chromatids. The sister chromatids have separated completely. The nuclear envelope again starts reappearing. See, the nuclear envelope has started reappearing. Then the cytoplasm is undergoing division and ultimately 2 individual cells are being formed. I hope you have understood it. I'll repeat the whole process once again. What is happening? I'll say once again. Listen very carefully. It is starting from the mitosis. Mitosis, interphase. In the interphase, what is happening? In the interphase, the chromosomes, they are replicating, they are dividing. Okay. Then what is happening? The nuclear envelope which is surrounding the chromosomes gradually starts disappearing or disintegrating. The chromosomes get organized along a middle plane, that is the equatorial plane. The 2 chromosomes get separated from one another, moves towards the 2 opposite poles, one towards the south pole, one in the north pole. Okay. Then what happens? The spindle apparatus gets attached to the chromosomes, to the center of the chromosomes, which are known as the centromere. Okay. Then what happens? Then gradually there is a pull from the opposite direction. This pull is because of the centromeric spindles. Okay. So these spindles are pulling apart the sister chromatids. Ultimately what happens is there is a complete separation of these chromatids, okay, leading to the again integration or formation of this nuclear envelope, and ultimately one particular cell gets divided into two daughter cells with the same number of chromosomes intact. So that is the basic function and the most important function which is performed by a centrosome. And another important thing I want you to remember is that not only these, after division, you will think that after division maybe only the chromatin threads form the chromosomes in the daughter cells. No, it is the centrosomes which are forming. It is the centrosomes along with the separated chromatins which are forming the chromosome structure of the daughter cells, right? Okay. Next, moving to our next slide. So, what are the other important functions of a centrosome? Basically, it serves as the center of production of microtubules. Okay. Next is it regulates the directional growth of certain different type of cells which are known as neurons. Another thing you should remember is they organize the primary cilia and flagella. That is, if I speak very specifically, these structures are responsible for forming the basal bodies of the cilia and the flagella. The basal bodies are formed by centrosomes. Okay. And another most important function you have already understood, that is the role that they play in cell division by the organization of the mitotic and the meiotic spindle apparatus, right? So with that we come to the end of this session. In the next class we will be studying about cilia and flagella which are helping in movements of a cell. Until then, stay safe. Thank you. And if you like this video, then please do share, like, subscribe and comment in the comment section below, and let me know any suggestion, anything that you would like to communicate to me. Anything would be appreciated. And till then, stay safe, and let's meet in our next session. Bye.